Thank you very much for the invitation. And thank you all for still being here after 2 full days of conferences. And I'm Maude Schneider. I work at the University of Geneva in Switzerland, and I just wanted to give you a little bit of background about myself, because I think that helps to explain why I'm doing what I'm doing at the moment. So my main background is clinical psychology, and I work in the field of neurodevelopmental disorders, mostly autism, and a specific genetic condition that I will talk to you about that is called 22q11.2 deletion syndrome. And so I did my PhD in Geneva, in Switzerland, and during my PhD, I was mostly working with clinical questionnaires and standardized tasks, and I was really asking myself what we are actually measuring with those tasks, and I was quite intrigued by that, and I wanted to go a bit deeper into more like ecological methods, and so I moved to Belgium for my postdoc. So I moved to the team of Professor Maude that you may have heard about. So she's one of the experts in the field of ecological momentary assessment. And so I conducted some studies with her, and then I came back to Switzerland. And so the goal was really to apply those kind of techniques to my population of interest, which is neurodevelopmental disorders. So what I'm mostly interested about is to understand why those conditions are more at risk for developmental health difficulties, and especially through the social processes mostly. So that's what I'm going to talk to you about during this presentation. And so my talk will be divided in 3 main parts. So just a brief intro about neurodevelopmental conditions and mental health. And then I will talk to you about EMA and how we can measure daily life social functioning with EMA. And then I will walk you through some of the results that we have conducted in our lab, in my lab, about daily life social functioning and mental health in populations. So probably you all know about autism spectrum disorder. We have heard a lot about it already. And just to specify that it's a neurodevelopmental condition with early onset of social difficulties. But what I think is quite striking in the literature is that there is really a paucity of studies that focused on the self perspective of individuals with autism and how they experience their social life actually. And what is also important to have in mind is that there is really an increased prevalence of mental health difficulties in young people with autism, especially during adolescence and adulthood. So this is just a small graph of the samples that we have collected in my lab, where you see that actually there is a whole range of mental health difficulties in those individuals, mostly mood and anxiety disorders, and that's typical of what is described in this population. That especially women with autism spectrum disorder have a higher risk of developing mental health difficulties. So we have, since a couple of years, collected a cohort of young people with ASD aged between 12 and 30 in my lab. They are mostly individuals without intellectual disabilities, so that's something to keep in mind. And mostly people who have received a relatively late diagnosis of autism, and they are mostly recruited through clinical centers, so which means that they have a lot of mental health comorbidities. So it's a sample that is enriched, I would say, for people who are quite challenging in terms of mental health. And so we have conducted this cohort where we have a bunch of measures, cognitive measures, clinical assessments, eye tracking, and EMA that I will talk about more. And another population that I will talk to you about is this specific genetic condition that is called 22q11.2 deletion syndrome, so 22q for short, which is a relatively rare neurogenetic condition, 1 in 1,000 to 1 in 4,000 live births, which is in most cases a de novo microdeletion, so it's a spontaneous deletion on the long arm of chromosome 22. And it's a condition that is associated with a whole range of also somatic manifestations like cardiac defect, velopharyngeal insufficiency, hypocalcemia, and so on and so forth. And so some of the children, especially those who have a heart condition, they are diagnosed quite early in life, sometimes in utero. And some of the children who have less somatic manifestation, they can be diagnosed later on. But what is especially interesting for us in this syndrome is that it's a genetic syndrome that is associated with a really increased rate of mental health difficulties and especially psychosis. And so for a long time, it has really been considered as some sort of a model for studying psychosis in a more homogeneous sample. And so this is a large study that we conducted now a while ago, international study where we combined information from different cohorts, and we found that between 30 and 40% of the individuals included in those studies were diagnosed with psychosis in adulthood. So this is a really increased risk of developing psychosis in this population. And in terms of social behavior, so that's also a very interesting population because we have children that are usually typically described as calm and socially inhibited, and but they are described as having a preserved social motivation, especially during childhood, but that tends to decrease around puberty. So they become more withdrawn, they develop also more social anxiety, and so they tend to be less isolated when time goes on. And there has been also some description of social cognitive impairments in this population, theory of mind, emotional cognition. They also have an atypical visual exploration of social stimuli, a bit similar to what is described also in autism. And there have been some studies suggesting that actually the social functioning impairments and the social cognitive deficits are associated with the presence of psychosis in this population. So in Geneva, we have a longitudinal cohort of children and individuals with this syndrome that started when I was not doing research at the time in 2001. By Professor Stefan Elias, and we are now coordinating this cohort together, where we have approximately 250 families involved, and that we follow up every 3 years since they are coming in the lab. And so for some of the participants that we have seen, we have almost a 20-year follow-up on these children, so which makes this cohort quite unique. And so in the cohort, we also have a large panel of measures like neuroimaging measures, behavioral measures, and we also introduced some EMA a couple of years ago. So basically, just take home from this first part is that autism and 22q, there are two neurodevelopmental conditions that are characterized by pronounced social impairments, especially during adolescence for 22q, and also high rates of mental health difficulties. But they seem to have like a different developmental trajectory in terms of their social impairments. So more early impairments in autism and later onset social impairments in 22q. And in those populations, we have some arguments to suggest that social impairments may be implicated in the development of mental health difficulties, but we don't know exactly how. And also, we don't know exactly how to capture relevant aspects of social functioning in those populations. And so this is why EMA comes in. And so you've heard about EMA just in the previous talks. And so the big advantages of EMA is that it allows for repeated assessment during several days and also in the context. So that's something that I think is very important. So you can also take some contextual information into account. And so you can capture also fluctuations in terms of mood and symptoms. And so in our protocol and all the studies that I will show you, we've used a similar protocol. So we have a 6-day protocols where participants, they receive 8 notifications per day. And so quite intensive protocol where they are asked about their affects, symptoms, and also some information about their social context. And what we try to divide it is to have more like quantitative aspects. So every time they receive a notification, they are asked to report whether they are currently with some people, yes or no, and with, if yes, with what type of people. So friends, parents, etc. And then we ask them more about their quality, their experience, their subjective experience of those social interactions. So if they are currently with other people, they are asked to report if this social interaction is pleasant, if this is stressful, if they feel threatened, those kind of questions. And they also are, if they are alone, they are asked to report about their subjective experience of being alone. And they are also asked whether they feel lonely or not. And why we differentiated between those more like quality and quantity aspect of social interactions is that actually when you try to link it with mental health, the more subjective and qualitative aspects of social experiences are important. So this is a study that was conducted by Robin Achterhoff in Leuven, and so not at all in neurodevelopmental disorders, but he did a study with an EMA protocol combined with self-reported assessment of mental health in a general population sample, and he tried to see what was actually related to the intensity of psychopathology in this sample. And what he saw was that the more like social quantity aspects were not at all related to mental health, but the subjective aspect of their social interaction was associated with mental health. So that was quite interesting for us to say, okay, we need to focus on those subjective aspects. But when we try to look at some of the measures that are typically used to assess social functioning, for example, the social functioning scale, they are actually more related to those quantitative aspects than those qualitative aspects. So that's why we thought that it was actually very relevant to use EMA to really focus on those subjective experiences. But when we started to do EMA in neurodevelopmental disorders, it was in 2018. There were almost no studies out there, so a few feasibility studies on like 10 patients or something like that. So we're really asking ourselves, okay, is it actually feasible to use EMA in those populations? Those are vulnerable populations, and especially during adolescence. So can we do it? And short answer is that yes, we can do it. It's completely feasible. There were a lot of studies conducted in other clinical populations, and so here just to show you that. We included our first 60 participants with neurodevelopmental disorders compared to 48 controls, and with our typical protocol, and we saw that we had a comparable compliance rate, that they didn't really show differences in terms of compliance. The only differences that we had is that they took a bit longer time to answer to each of the questionnaires. So that's something that we had to take into account, that to reduce the burden of the questionnaires, to have a survey with not too many questions involved, because it was taking a bit more time. And so the take-home message for this second part is that the quality of social experience is especially important and related to mental health compared to more quantitative aspects. And EMA is a relevant technique to assess quantitative and subjective aspects of social functioning, and that it's possible to actually implement EMA protocols in youth with neurodevelopmental conditions. So what we do is, with those things in mind, is that we started to try to characterize daily life social functioning in those populations. So this is one of the first studies that we conducted, where we try to indeed just characterize the social quantity and the social quality in those populations. And here, what you saw is just the percentage of time that people were spending alone. So you can see that in blue. And the percentage of time that people were spending, lost some of my legends. So this is alone, with family members, with peers, and with strangers. So what you see, so the first result that was actually quite interesting for us is that typically we say that young people with mental conditions, they are socially isolated. So that's true, yes and no, because that's not true in the sense that they don't spend more time alone than young people from the general population, but they have some sort of a different involvement in the social sphere, meaning that they are spending more time with family members, so parents, siblings, and less time with peers compared to young people from the general population. So they have a different involvement in the social sphere. And when we look at the more like subjective aspects of the social experience, what you can see is just the comparisons between the different items that we use. So whether they would like to be alone when they are interacting with others, whether they like to be with others, if they felt judged in the company of other people, or if they felt nervous. And what we can see is that, interestingly, our young people with 22q, they had a similar experience of social interactions than our control group. But the young people with autism, they have a more like negative experience of social interactions, so subjectively rated as less pleasant, more stressful, and the only item where both clinical groups were different from the control was this one, which meant that when they were interacting with others, they reported that they would like to be alone to a greater extent. So we interpreted this as a significant cost of social interactions, that it was actually costly for them to be interacting with others. And when we were focusing on time spent alone, we saw that actually all the three groups reported that they were feeling more alone, more lonely, when they were actually alone than compared to when they were with other people, which makes sense. But there was no between group differences in terms of the density of the reported state loneliness or the experience of being alone. But so our second question was that, okay, are there some of these indicators associated with mental health in those conditions? And so we looked at what we called affective reactivity to stress, and especially to social stress. So which means how much your negative effects increase when you experience social stress. So when you are experiencing that you are in a stressful social situation, your negative effects tend to go up. So this is the case. So this is an example in 22q. So this is the case in the control. So when your social stress goes up, your negative effects goes up. And this was the case as well for young people with 22q. So, but on average, there was no significant group difference. So there was not like an increased reactivity to social stress in 22q. But we could see, I don't know if you see in the back, that there was a large inter-individual variability between individuals within 22q, which was actually relevant because when we looked at this inter-individual variability, we saw that those who were more reactive to social stress, there were also those who reported more severe psychotic symptoms in the context of 22q. And it was the same thing when we looked at loneliness. I told you that there was no between group differences, but when we looked at the inter-individual differences within each group, we saw that actually more intense feelings of loneliness were associated with self-reported mental health difficulties, both in the context of 22q and in the context of ESD. So basically that suggests that the distress associated with social interactions, whether you are in a social context or whether you are alone, is actually something that is relevant when we are talking about mental health, because that's what seems to be associated with mental health difficulties and psychotic symptoms, especially in 22q. And so this is a very complicated slide. You don't need to understand everything on it, but something that we are trying to do at the moment is to go more into like multivariate analysis and more dynamic symptom profile of EMA. Because here what you saw is that we took one variable at the time and we try to correlate it with mental health. So this is interesting to look at specific association, but I think what is missing at the moment is to have like a broader pattern of what influences what in a more like network analysis way. And so this is something that we are currently working on with Corrado Sondini, who is a medical doctor at the University of Geneva, and who got a grant specifically to develop those aspects. And what we are trying to do is to use our EMA symptoms within a network, and to use that also to try to study the dynamic of each from one time point to the other, and to use this as layers. So if you see the dark green here, for example, that would be one network of EMA experience, momentary experiences, and how this is related to the next network of momentary experiences, for example, 3 hours later. And to try to see how this is actually different between the groups, and also what are the relevant, in a way, pathways linking one moment to the other. So this is something that we are currently working on that is not published yet. But what is actually interesting is that we can try, because Corrado has also a neuroimaging background, so he tried to implement methods that are taken from the neuroimaging field, and especially he tried to develop algorithms based on the tractography measures to try to extract some bundles of relevant paths between one time point to the next, and to see how those bundles could actually differ across population. And this is just probably don't see anything, but you have to trust. You have to trust me. That actually what is quite interesting in 22q preliminary findings that we have is that actually there is a significant bundle linking social distress, so feeling actually stressed in social situations, with the fact that they tend to experience psychotic experiences at the next time point. So that seems to be something that is quite specific to this group, and that is for us quite interesting, because we would have such a more dynamic perspective of these associations between social stress and the emergence of mental health difficulties. Mm-hmm. And so this is my take-home message for this third part, is that we can extract from EMA indicators of social quantity and social quality. And when we compare 22q and ASD, we can see that they share some similarities. So, for example, in terms of social quantity, they are quite similar, but they have different social experiences. And so they have different social profiles and different social needs. And what is interesting is that in ASD, what we found is that they have overall a more negative subjective social experiences in their daily life. And in 22q, compared to what we were expecting, actually, they have subjective indicators that, on average, are not different from what is reported in typically developing peers on average. But we see large inter-individual variability that is meaningful, because when we look at this inter-individual variability, we see that the distress that is related to social interactions is actually related to the presence or the severity of mental health difficulties. And yeah, that's what I was telling you later on, is that we need, I think, more multivariate and more dynamic approach to study those phenomena actually. And to just give you a bit of perspective, what we are quite interested in at the moment is to study also the contribution to the exposure to adverse interpersonal events, such as bullying or experiences of victimization. And because we have some arguments to say that actually there is an association between exposure to adverse events and psychotic manifestations in 22q, for example. But in the previous studies that we conducted, we didn't really focus on those interpersonal stressors that I think are especially relevant. And so we have a project that will start next year, where we will actually focus on those adverse interpersonal events in young people with neurodevelopmental conditions, where we are actually want to study the link between those adverse interpersonal events and mental health through changes in the affective and the physiological reactivity to minor interpersonal stressors. And we would like also to study the impact of more like regulatory functions, such as social cognition and attentional processes, to see whether that modulates this association. And just quickly finish on that. So we have some arguments to say that there are actually dysregulations of the stress system in those two populations, because we found alterations in cortisol levels. We also have like preliminary findings in heart rate variability that is decreased in autism, and that is associated also with mental health difficulties. And we also have like preliminary findings in autism linking this exposure to interpersonal stress with more attentional biases towards threatening information. That is, I think, also quite interesting. So I will be happy to come back to present you some of these new works later on. And with that, I think I would like to thank you for your attention. And I would like to thank the lab of Professor Stefan Elias, with whom I'm collaborating quite closely, and also my lab and the PhD students who conducted most of this work. Thank you very much.